say thank you first to the organizers and for everyone who's here. It's a very interesting group to talk to. I actually already learned a lot in just an hour or 2 hours or so this morning. And I have to apologize for the title. I don't mean this in a bad way, that the omics is just crazy and it's leading us in the wrong direction. By no means is that the case. I think it's very impressive what we can do in terms of the amount of data that can be collected and the diversity of the data from genomics, proteomics, metabolomics. The question is how to put it together, obviously, and make some sense out of it. So I have an example on that to show you the second part of the talk. The first part, actually, I have to back up again. I'm not really, although I've been doing mass spectrometry-based analysis for 20 or so years, I don't consider myself an analytical chemist. My background is more nutrition, biochemistry, and a lot of stable isotope tracer-based analysis. So we use the mass spec as one way of probing whether something is turning over or not. NMR works just as well. It has its pros and cons associated. But by way of introduction to the talk, I don't need to tell you guys, omics allow you to cast a big net, whether that's by NMR, it's mass spec, it's whatever. You're throwing a big net out there and you're looking for differences. And really, I could say at a high level, the central goal is, are there differences between groups, something up or down? And we're not really, as I understand it, with just a static profiling, you're not really getting at the mechanism of the differences. So the first thing we've been trying, or I'll try to talk about, one of the things we're really trying to do a lot, especially at Merck, is could we use isotope tracers in these omic analysis? So not exactly like what Chris was talking about, but what we want to do is put the tracer in and watch the isotope pattern change over time. And from that, we can infer something about the actual rate of metabolism. And the second part of the talk is really to consider the impact of the data normalization on what we think is normal or abnormal. So again, I don't mean to trivialize this when I say lipidomic profiling is easy using LC-MS. In principle, it's easy to collect the data. And I have to say I don't want to endorse or whatever any instruments in our lab. I'm actually very fortunate to have a range of equipment from Agilent to Thermo to Waters. And this is just one example of a lipid profile we collect on this Waters Synapt instrument. And the spectra looks, or the chromatogram may look kind of clean. In 10 minutes, you get just looks like a few different peaks, but actually it's a very complex spectra. Here, there's several hundred different lipids that you can find under here. So it's relatively easy to collect the data. In a few minutes, you can collect several hundred or 1,000 different analytes. If you break this down into another example, this is just a profile of cholesterol esters. And so you can easily see when you look closely in this retention time and you look at the different, I don't show you the masses, but you have 18 carbons with 2 double bonds, 16 with 1 double bond, so on and so forth. So this is lipidomic, typical lipidomic profiling. You could tell something about what cholesterol esters are there and how abundant might they be. The question that we're faced with sometimes in the drug discovery side of things, and obviously you guys have your own examples of this, is that if we're after, for example, understanding whether this is cholesterol palmitate, is it different between a control and some treatment? It's up or it's down, whatever it may be. As an analytical person, that's kind of easy to think about it. Oh, just give me the sample, I'll put my standard in, I'll see if it's different or not. But you have to appreciate that cholesterol can actually come directly from the diet, just as palmitate can come directly from the diet, or the diet can actually give you, sugar, and that can be synthesized into palmitate or cholesterol. So although this is a relatively simple molecule, you can think of it as having 4 parts. The palmitate can come de novo synthesized versus the diet. The cholesterol can come from the de novo synthesis or the diet. So when we're asked a question, we're trying to answer a question, is cholesterol palmitate different? It could be the static profile, it could be the kinetics. And so one of the ways we try to approach the kinetics is we use a lot of deuterium labeled water. And this is just a kind of simple cartoon that shows acetyl-CoA, so this molecule here is the carbon precursor, if you will, for palmitate or for cholesterol. And those hydrogens that are bound there eventually get incorporated into the lipid you want. NADPH is used as a reducing source, and there's hydrogen from water that comes in. So if you actually expose an animal, a human for that matter, to some small amount of deuterium-labeled water, you can watch over time the deuterium pile up in the lipids, and from that you can infer something about the synthesis rate. So we did a very simple experiment at Merck where we gave normal mice, and we fed them either a diet that basically had all sugar in it, or basically no sugar. It was almost all fat. It's a ketogenic diet, people in nutrition would call it. And these animals were given a single injection of deuterium-labeled water, and then we take the samples after a certain amount of time out to about a week or so. We like to work on Monday, Tuesday, Wednesday, Thursday, Friday, and we take Saturday and Sunday off, so we only collect days 1 through 4 here. And we put this cholesterol palmitate into the LC-MS-MS instrument, and you can see very clearly, if you look at the labeling here, and this actually is the M+1 isotope relative to the M0 isotope. I'm going to show you a cartoon of that in a minute. You can very clearly see that animals who eat a lot of sugar, they seem to make a lot of this cholesterol palmitate, and animals who eat no sugar, they don't seem to make very much of this cholesterol palmitate. So that's somewhat intriguing, but it leaves you with a question, well, where is it, right? Where is the label? Are they making the fatty acid and not the cholesterol, vice versa? What we're able to do actually with this LC-MS-MS is that we acquired in this MS to the E mode. So we'll run one scan where we acquire a full spectra, and then the next scan will acquire a fragment spectra. And so in one run, you get a full scan to know something about the molecular weight of the compound, and then the second scan, you get a fragmentation spectra, and then you stitch these things back together after. And when you look at the actual fragment, in this case, we don't get a fragment that keeps the palmitate with the fragment ion actually contains just the cholesterol. When you look at the cholesterol side of this molecule, you see a very different picture now, actually. So what you can now conclude is that the difference between these two spectra is actually the palmitate labeling. So we can go back now and put the model together, and we can say, look, when the animals eat a lot of sugar in the diet, they're able to actually drive this part of the pathway, fatty acid synthesis, but the sugar content of the diet really didn't have much effect on the cholesterol synthesis. If anything, it might have had slightly the opposite effect. So the mice ate more and more sugar, they actually activated this part of the pathway, and as they ate a little bit more fat, they actually seemed to stimulate this. And this is not to drive home the message that sugar is absolutely going to do this, because the type of sugar and other things in the diet might play a role. It's just an example of how you can use the tracers at the same time you're doing a metabolomics to do this sort of flux omics and pull these images apart if you want. And what we've done other studies is if you put just a little bit of cholesterol in the diet, actually, you can really have a big effect on the cholesterol arm. So now you can actually get pretty comprehensive kinetic phenotyping, if you will. The problem we run into, though, is that, or I guess the pro and the con of using the tracers is that you can always make the customer happy. If the customer comes to you and they say, the customer, one of our biologist collaborators, we want to know whether something is different. If all we had was the static phenotype, well, we might make some people happy and we might not make others, because they might be different, they might not be different. If we have the option to get a kinetic phenotype, maybe we can make everybody happy, because now you have more chance of finding differences. So here's one example where, again, animals were eating this sugar diet, or basically this fat diet, and we looked at the phospholipid profile. This is a phospholipid that has 34 carbons with 2 double bonds. And if you just look at the peak intensities, there's no difference. So if the customer wanted, our colleague wanted to know, is there a difference in phospholipid metabolism based on sort of lipidomics or static profiling, we'd say no. But if you looked at the kinetics of this, these are, again, the same model, they were given labeled water, you would see very clearly that they're very different, that although the concentration of this phospholipid isn't different, the fatty acids are all coming by the de novo pathway versus the other that are not. And then obviously you can get these counterintuitive profiles where this is a different kind of triglyceride. It has 54 carbons with 4 double bonds. And in this case, it's very clear that the static profile is different. In animals that eat the sugar diet, they have very low concentration of this. When they eat a lot of fat, the concentration goes up. But this doesn't really tell you anything about where the fatty acids are coming from. If we go and we look at the spectra, and we look at the change in the isotope profile over time, it's very clear that in this diet, although the concentration in the blood is low, actually the fatty acids are all coming from this de novo synthesis pathway. The spectra in the middle just shows you one example of what it looks like, the complexity kind of these spectra when you collect the final time point. But what we're really looking at in this curve is this shift in the time course. I'll try to show you that in a second here. Actually, I'll come back to that in a second. Try to summarize a little bit where we are now, because you've heard examples of giving labeled CO2, giving labeled glucose, and now I'm telling you about giving labeled water. One of the reasons we like to give the labeled water, and there's no objection to giving the other ones, it just depends on the model that you're working with. When you're dealing with these living animals, although the blood glucose, for example, may not change much over time, the concentration, the half-life in people is maybe about 20 minutes. So if you give someone a tracer of glucose now, in 20 minutes or say in an hour, it's all gone. So if you have some end product that's really rapidly being turned over and synthesized, well, you can probably give them a shot of labeled glucose and you can watch it, the fatty acids get synthesized. But in other cases, it's not really practical to do that. So this is an example where we tried to do a very comprehensive dissection of a pathway in terms of the cardiovascular disease. We're very interested in manipulating blood cholesterol levels, and you can do this by converting them to bile acids, and cholesterol comes from a couple different places. Either you have de novo synthesis or you have this reverse cholesterol transport process. So our question was very simple. If we give this compound called cholestyramine, which is known to pull the bile acids, and it's known to stimulate this, would we actually affect this, this reverse cholesterol pathway? So this is a very comprehensive systems biology-like thing. We're doing one perturbation here. We want to know if we're manipulating lipid pathways and protein pathways that are related. To do this in vivo is a little bit tricky. And this is sort of the take-home message here of how some of this works. So what we do is, again, we give the animal a dose of deuterium-labeled water. And Chris's example shows that his cells are either 5% enriched or 95% enriched. What we're taking the animal to here with the labeled water is only about 2% enrichment in their body water. So it's just to put it in perspective for you guys. But the nice thing is that you see the half-life here. This is the labeling of body water, and this is a mouse. It stays around for several days. It has a half-life of about 2 days. So when you have kinetics that are relatively slow, something doesn't get synthesized very fast, or you want to know a sort of integrative picture, what happens between the feeding and the fasting transition, you really want to have a tracer that hangs around for a long time. So this is very nice. This is the precursor, and then you can watch labels show up into cholesterol, into cholic acid, or into the apoproteins, and we can fit these curves and we can infer something about this metabolic network in terms of the kinetics. But what this has come down to is sort of the nuts and bolts of how the mass, at least in our end, how the mass spec works. So you've seen this kind of picture before. This would be the monoisotopic mass of, I don't know, some compound. Maybe this is some big cholesterol ester. And then there's a bunch of carbons, so each one has a 1% chance of being C13 labeled. So you have an M+1 signal, an M+2, and so on and so forth. What we're trying to do is a little bit different than the example you guys saw before, where the mass profile really shifts and you get a new profile that starts way out here at the M+4 or whatever it is. What we're doing is actually watching very small shifts in these profiles, in the ratio of the M1 and M2. So the question comes down to how sensitive is the mass spec to picking up those, or precise anyway, is the mass spec to picking up those small shifts, especially when you acquire data in an untargeted mode where you're scanning the mass range relatively fast. So this is an example of the CV of this ratio of M+1 to M0, whether we're looking at these phospholipids, different phospholipids, or whether we're looking at triglycerides. And this is actually data acquired from a plasma sample, and where there's no spiked in deuterium or anything like this. It's just natural background. So we expect the theoretical ratio, for example, on one of these, I'll pick up this one, this triglyceride, the theoretical ratio is about 60%. So this signal is about almost what I have here at this example. And what we're able to do is actually measure that thing with the reproducibility of about a 1% or so CV. So if the background ratio was supposed to be about 50%, the mass spectrometer actually can tell you it's 50 plus or minus 0.5. It's actually that tight. And this is using the commercially available software. We haven't fiddled around with anything like that. So it's actually pretty reproducible. I'll come back to that in a minute. And so the second part of the talk, and this will be quick, only a couple slides here, is to consider the impact of data normalization on the interpretation. So this completely shifts gears. This part is not really on tracers per se, but it takes me back actually to work I did a few years ago before I was at Merck, where we were interested in this question about adrenergic resistance, and is it a cause or a consequence of lipid accumulation, obesity, adiposity, whatever you want to call it. And so this little cartoon gives you an idea of what's going on. Triglycerides are found in adipose tissue. Epinephrine is one of the hormones that can actually affect the breakdown of these things. So the question is, do cells become resistant to epinephrine? And that leads to the accumulation of lipid. So we ask a very basic question. We took normal mice, we fed them a normal diet, or we fed them this high-fat diet, and after a certain amount of time, we took these epididymal fat pads out, we digested them, and we did studies on isolated cells. where basically we took those isolated cells and we added different amounts of epinephrine and we looked at the glycerol release to ask this question. Is there an effect on lipolysis or not? Is there a resistance that occurs? And so again, you know, we're in the business of making everyone happy. You can pick whichever plot you want here and we all go home happy. The first plot, and this all comes to playing with data normalization. The first plot would be the image you'd get if you actually did this study in vivo. And what people normally do in vivo is they would say, look, there's a certain amount of glycerol released for the different concentration of epinephrine. And in a control, that's the dose response that you get. In an obese, that's the dose response you get. And people might conclude, oh, there's actually resistance, because typically they might normalize the data as glycerol released per milligram of triglyceride, per mass of the fat that's actually there. By doing this study in the isolated cells, we actually have different options now of normalizing the data. So in this panel, it's the exact same data we have here, except we normalize to micrograms of DNA. So we ask a different question. How is the actual cell itself responding to the stimuli that we have? And when we did that, we concluded that actually the cell itself didn't seem to be disrupted in terms of its metabolism. The cell was working well. The data normalization here is because we normalize against the amount of lipid that's actually there. And then the last figure, which this one actually takes the take-home message to the audience here, will come on the next slide, is that by doing it in a cell-based study, we could actually say we know the total amount of DNA that was there. We know the total mass of cells that were there. So we could actually normalize the data as nanomoles of triglyceride that's burned or released. And there actually, the spectra is, or the profile is actually completely flipped from what it was before. So because you have more cells, the same dose of epinephrine has actually a larger effect. And so this comes to the question that, actually more question to pose for discussion, is that when is the case of normal actually abnormal? And so the scenario I just described for you is that, look, if I have measured my epinephrine concentrations in a healthy scenario versus an obese scenario, and I do whatever analysis I have, and I conclude that the concentrations are the same, does that mean that healthy is the same as obese? I would say probably not, because what we're not considering in this analysis, in this interpretation, is that where's the target effect of this thing? And so the concentrations might actually be the same here, but if the mode of action is to act here, and you actually have more of this target, well, there's actually probably a physiological difference in spite of the fact that analytically they're identical. And so you could flip this a lot of different ways. You can substitute insulin here if you guys follow diabetes, and the same thing. So I would say actually, in order to be normal, what you might want to have is actually a higher concentration of epinephrine in the healthy versus the obese, because then you get less of an effect on the lipid release. And the opposite for insulin, you'd actually, as adipose tissue increases, you'd actually want to have more insulin here to help control this reaction. And as I said, this is a weird way of playing around with the data. It's a unique way because you do the study in vitro. You can do this, but it made us stop and think a little bit. So with that, I conclude that I think actually, as you guys probably are well aware now, it's very easy to get a lot of very good data out of the Omic type of analysis. I think putting the attention on isotope labeling patterns is fantastic. Our experience is actually that the hardware is generally much better than any of the commercially available software that's out there, and developing novel software is clearly the way to go. And the second part is that there's not really a right or wrong answer for this data interpretation. It's just obviously an example where it's easy to be fooled by some changes in body compositions and so forth, depending on someone's application. And with that, I'll conclude, and I have to thank, actually, I started at Merck about 4.5 years ago. I'd been at Case Western before that, and shortly after I was hired, we hired Jose Castro-Perez, who was at Waters, a demo chemist at Waters. So he's really responsible for setting up a lot of the methods in the lipidomic profiling and allowed me to plug in some of the kinetics to that. With that, thank you guys.
Very nice presentation. Very exciting data and information about this. What I would like to, I don't have questions, I have a comment. A comment when, you know, the metabolomics is a tool of measure things. And it's getting more and more sophisticated, accurate, and blah, 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 blah. However, when you try to use a tool, the first thing to do, you have to go back to study design. Mm-hmm. When you define your study design and you know, anticipate all those things, then you might have an answer. You know, as well as statistics, which can flip those depending on normalization protocol, median or, you know, total line or whatever. Yeah, That's happening for the low abundant metabolites more frequently than for high abundant. But basically, when we observe situation like you described, there's very nice options to go back to study design. And look, is this the answer which give you another question can be resolved?
Yeah.
So you might need to run another experiment.
Oh yeah, I think there's no doubt. Actually, you get one question, gives you an answer, gives you a new question.
That's right.
That's for sure. The one thing I didn't comment on here, actually, the one I'd say somewhat advantage we usually have with the studies we're doing is we're actually very fortunate. We're not asked to go in and do untargeted things and hypothesis generating. Usually someone has a knockout of some kind of model or a compound and they ask us, it's very specific. So then you can kind of build in advance, within reason, what you really want to get high priority data on and so on and so forth. So although we acquire a lot of this stuff, like in this omics untargeted fashion, it's very rare actually that we use it because in the turnaround time for the understanding the data and so forth, it's definitely too slow for us.
Yeah, so. You're right. But basically, there's not much difference between non-targeted and targeted. All the statistics manipulation works for both of them, just cleaning up the data.
Yeah.
But the question else, you see, there's too many confounding. For this particular experiment, there's too many confounding parameters since the very beginning. And then you try to use statistics to introduce, okay, what we normalize to the weight of the mouse. Are they just obese that just get bigger? And things like this. I'm not going to go for detail. It is a very complicated case, but it may be resolved.
Yeah. Thank you. Thank you.